Hi, Dr. A again. So we're going to talk about the urinary system this time. So the urinary system consists of your two kidneys that filter the blood, then the two ureters that lead from the kidney down to the bladder. Of course, it consists of the bladder, a urethra to convey the nitrogenous waste substances to the outside, and that's it. So the urethra is the exit, if you will. It helps maintain normal water concentrations of electrolytes in water. That's its main job. It regulates pH and fluid volume. That's also one of its main jobs. And it helps control red blood cell production and blood pressure. So here they are. So you can see the two kidneys here, right? So like right below where your rib cage is in, right there. The ureters lead from the kidneys, the bladder, and the urethra there. Okay, so the kidney is a reddish-brown, bean-shaped organ. It's about 12 centimeters long, so probably close to the size of your hand, probably a little bit smaller though. It is enclosed in a tough, fibrous capsule. So here in this cadaver dissection, they're in green. They're just highlighted in green, but normally they would be that like dark brown, really kidney bean color. So they are positioned what we call retroperitoneally on either side of the vertebral column, with the left kidney slightly higher than the right. So what that means is basically all the way in the back of your peritoneal cavity. So on top of your kidneys, you would have your other, your digestive system organs and all of that. Reproductive organs would be over here and all of that. So they're at the bottom, right? As you look down into this cadaver. Then connective tissue and adipose tissue do hold the kidneys in place. And there is a medial depression. So this is on the medial surface. Medial is towards the middle, right? And it's called the hilum. Right there's that depression right there is the hilum. And it leads inside to a hollow renal sinus in which the blood vessels, nerves, lymphatic vessels, and the ureter enter. So everything basically comes in or exits out of the kidney through in the area that's the hilum with the renal sinus and stuff. The kidneys, so if you look at a cross section of a kidney, we're going to investigate here. So inside the renal sinus is a renal pelvis. So here, this renal sinus is this cavity right here, right? And here is the renal pelvis. And so it's just think of it as like a big funnel that leads over to the ureters. Sorry, I'm going to pause this for a second. Okay, so sorry about that interruption. I have a cat at the vet currently. So we have the renal sinus, which is, again, this cavity here. And then we have the renal pelvis. And this renal pelvis, so we definitely have this one funnel, but then you have this right here. All of this is part of the renal pelvis, but it's basically, these are subdivisions that are like mini funnels that are funneling into larger funnels that then funnel into the largest funnel. And so these are called calyces. So this is a minor calyx. So underneath each pyramid, you have a minor calyx right there. So it's a funnel catching all the little drops of urine being made right there. And then, for example, here where two minor calyces join, you have a major calyx right there, right? And so just, it's kind of like a branching system of funnels where eventually it all ends into this one big funnel, and then that goes down to ureter. So there are two distinct regions that are found within the kidney, the renal medulla and the renal cortex. So the medulla is here. It has the renal pyramids, and it leads to these papillae. So the pyramids are right here. So this all right here is the renal medulla right there. Okay. And then the renal cortex, it surrounds the medulla, and it's this outermost portion, and also dips in between these renal pyramids. So it's this lighter colored material here. This is where all the nephrons actually are. It's all of this right here. All of that's the cortex. So if you go around all the outside here, that's the cortex. And then the functional unit of a kidney is a nephron. And we're going to look into what nephrons are here in just a second. So again, if you here is, again, your renal sinus with the renal pelvis, minor, and then major calyces there. We have pyramids, we have the medulla here, well, pyramids are the cortex here. And if you took a slice of this, you would see it would look like that. So again, this region right here would be the cortex. This region right here would be a pyramid. And these are collecting ducts that are hanging, that are in here. Those collecting ducts gather urine and send it through past the papilla into the minor calyx. There are also portions of the nephron, so the ascending and descending loops are also dipping here into the pyramids. And then here are the main parts of the nephrons, which are where the renal corpuscles and the convoluted tubules are located. So this part of the nephron is in here in your renal cortex. Okay, so in here, I say circle, you can actually trace, if you will, the renal pelvis. So again, you just select a color. It doesn't really matter what color you want to use, thicker, thinner, and then draw on the picture and circle it and then submit it. Same thing for here. You need to circle a minor calyx. So look in here and find a minor calyx, and you can, circle it or highlight it or, somehow indicate where it's at. And then don't forget to hit submit. And then circle a pyramid. So again, select a color and then circle it and hit submit. All right, so the kidneys function to regulate the volume, composition, and pH of body fluids and remove metabolic waste from the blood in the process. So urine is waste, and we're constantly producing it. The kidneys also have control of the rate of red cell formation by secreting erythropoietin, and erythropoietin is a hormone that regulates the formation of red cells. Okay, it also regulates blood pressure and volume by secreting renin. Now, we talked a little bit about, mentioned erythropoietin in the blood chapter because it's what stimulates the production of red cells. What triggers the release of erythropoietin is the kidneys sensing low amounts of oxygen. And if it senses low amount of oxygen, it's basically assuming there's not enough red cells to carry the oxygen that's available, and it will go trigger the release of erythropoietin so that you can make more red cells to carry more oxygen and deliver more oxygen to the kidneys and stuff. And then it will see, okay, we have adequate amount now. So it regulates blood pressure and blood volume by secreting renin, and we're going to look into what renin does here in just a minute. And then it also plays a role in activation of vitamin D, the sunshine vitamin, which allows you to absorb calcium. It has function in your immune system, to strengthen your bones and stuff like that. Okay, so let's look at a little bit at the renal blood vessels. So your abdominal aorta, which runs down your abdomen there, it has two branches off of it, and those are your renal arteries. And each renal artery, right and left, will enter its kidney, right and left, right? And as these arteries pass into the kidneys, then they, of course, branch off into smaller arteries. And those are your interlobar arteries, your arcuate arteries, cortical radiate arteries, or also interlobular arteries, and then eventually the afferent arterioles, which lead to the nephrons, and the nephrons do the filtering part. And then some of that, after the afferent, there's an efferent arteriole, and then it wraps around the capillaries, and then eventually, as it wraps around you're gonna see the tubular system. It turns into venous blood, and the venous blood then is returned through a series of vessels that generally basically follow the arterial pathway just backwards, right? And then the renal veins join the inferior vena cava and the abdominal cavity. So a kidney contains 1 million nephrons. That's a lot. If you have two kidneys, you have 2 million nephrons. Each of of which consists of a renal corpuscle and a renal tubule. And by the way, it actually takes quite a bit of damage to your nephrons before you really start having signs and symptoms of having, some degree of kidney failure or kidney involvement in a disease process. The renal corpuscle is the filtering portion of the nephron. It is made of a ball of capillaries that is called the glomerulus, and a glomerular capsule that receives that filtrate. So again, at that point in time, it is not urine yet. It's just a filtrate of the blood. The renal tubule collects that filtrate and leads away from the glomerular capsule. And first it becomes what we call the proximal convoluted tubule. And then it leads to the nephron loop, so the ascending and descending nephron limbs of the nephron loop. And finally to the distal convoluted tubule, and then each distal convoluted tubules will join the collecting duct, and the collecting duct does hook up to quite a few different distal convoluted tubules, and those collecting ducts pass into the medulla, and then they empty into a minor calyx at the papilla. So this again is what one nephron looks like. So here, this is the glomerulus. So here is the ball of capillaries. And this is the beginning of urine formation. So as it pushes fluid out of the capillaries, that fluid becomes a filtrate and is captured by this glomerular capsule. Okay, and that capsule captures that filtrate and sends it down here to tubule. So this is a proximal convoluted tubule, and you can see how there's a blood supply around it. And then we go down the descending loop, and then ascending loop. So descending limb, ascending limb of the nephron loop here. And then we have the distal convoluted tubule going on here. And then we hook here into the collecting duct. And you can see there's other hook area where this would be another nephron, another nephron, another nephron, another nephron. And then if you look at the blood supply within here, this one nephron, so you had a cortical radiate artery, and then it branches into afferent arteriole next to glomerulus, and then there is the capillaries that are wrapped around the convoluted tubules. These are called peritubular capillaries. And here you can see in this section here, all of this is arterial, but then this here is arterial, then turns to venous, and then venous here is going to return. All right, and then this collecting duct would empty right here at the papilla, and then it would be a minor calyx below that. Okay, so in this drawing, circle the glomerulus. So if you need to move this, just click that, and it'll reveal the whole drawing. So circle the glomerulus in this drawing, and then circle the distal convoluted tubule. Okay, so follow the path and find the distal convoluted tubule. Okay. And so again, the blood supply of the nephron, the glomerulus receives the blood from a fairly large afferent arteriole, and then it passes into a smaller efferent arteriole. So this is actually quite important. So it's illustrated here with the larger afferent, the ball of capillaries, and then the smaller efferent. Okay, so first of all, we mentioned this already, but afferent and efferent sound a lot alike. Afferent is A for arrival, and efferent is E for exit. So the blood arrives at the ball of capillaries and then exits here through the efferent. So afferent and efferent. Now, the efferent arteriole then gives rise to this peritubular capillary system right there, which surrounds the renal tubule right there, and it becomes eventually venous system. Now, this diameter difference is important. I want you to note it because we're going to talk about how that can help generate pressure, filtration pressure. So in this diagram, then find me the afferent arteriole. So just highlight the afferent arteriole based on what we just talked about. Okay, the next thing is the juxtaglomerular apparatus. So it's like, what? Juxta means next to. And then glomerular is the glomerulus. So it's a little area, it's a sensor area that is right next to the glomerulus. So there's a point of contact between the afferent arteriole, the efferent arteriole, and the distal convoluted tubule. And also, and with the epithelial cells of the distal tubule from the macula densa. So all of these together, those are all the components, if you will, of that sensor. And near the macula densa, on the afferent arteriole are smooth muscle cells called juxtaglomerular cells. And they have a really big role. So the macula densa, with the juxtaglomerular cells, they are the components, again, of that sensor, make up the juxtaglomerular apparatus. And they have a big role in urine formation and filtration and even blood pressure and things like that. So here's what it looks like. So again, here's your illustration of a nephron with a glomerulus and a glomerular capsule, proximal convoluted tubule, descending loop, ascending loop, distal convoluted tubule. So as you can see here, the distal convoluted tubule comes near the glomerulus, and if you zoom in, then we have our large afferent arteriole, our smaller efferent arteriole, and then we have the macula densa, and then the juxtaglomerular cells are right here. They're smooth muscle cells that are right here, wrapped around this afferent arteriole. Okay, and together, this sensory system here makes the juxtaglomerular apparatus. Okay, so we'll circle back to this in a second. Okay, urine formation. So urine formation involves 3 steps: glomerular filtration, tubular reabsorption, and tubular secretion. Glomerular filtration, the urine formation begins when the fluid portion of the blood is filtered by the glomerulus and enters the glomerular capsule as what we call glomerular filtrate. And if you will, it's a filtrate of the plasma portion of the blood, because the cells, like red cells and white cells, stay behind in the blood, and then really large proteins stay behind in the blood. This produces about 180 liters of fluid per day. Now, just so you can wrap your mind around that, just imagine your 2-liter bottles of soda, right? 90 of those. Okay, so like a big old shelf full. And so obviously we don't pee that much, thank goodness. We probably pee about 2 liters a day in an entire day, not at one time. So most of it obviously is returned to the blood through the process of tubular reabsorption. And then the last step is called tubular secretion. It's a way for your body to dump additional substances in the blood that maybe did not make it out through filtration, and to get rid of some things. So certain drugs can be dumped that way through tubular secretion. Okay, so let's start with step 1, glomerular filtration. So water and certain dissolved substances then are filtered out of the glomerular capillaries, and they're captured by the glomerular capsule. The glomerular capillaries have many fenestrations. Those are windows. So it's from the French word for window, which is fenêtre. And so there are little openings, little windows in those capillaries. And so the glomerular capillaries will allow more filtration than other capillaries. They're just more permeable. They allow more stuff to go through. Okay. But then there are cells that are called podocytes that are wrapped around these glomerular capillaries, and they help prevent the plasma proteins from being filtered out of the blood. So the big plasma proteins are supposed to stay behind. You're not supposed to have a bunch of protein in your urine. Okay. So what leaves your the plasma? Mostly it's water, electrolytes, glucose, urea, and uric acid. And again, your protein should stay in your blood, your red cells stay in your blood, your white cells stay in your blood. They are not part of the filtrate. So again, here's a close-up illustration of this glomerulus. So this ball of capillaries. So we have our afferent arteriole, our smaller efferent arteriole, and we have this ball of capillaries, and then these windows opening, these fenestrations, right? So it's quite porous, so it's easy for the fluid to leave there. But then you have this layer of podocytes that are wrapped around it, that are like a secondary filter, if you will, that just makes sure that the big proteins, albumin and stuff like that don't leak out of, uh, of, of this, from the capillaries and end up in the filtrate. So then, knowing that, what do you think would happen if those podocytes got destroyed or damaged? And so that's an interesting question, because that's what happens in, for example, renal failure due to diabetes, especially type 2 diabetes, is there's damage to these podocytes, and it causes certain symptoms and certain things to happen. So see if you can noodle that and think that through. So just to give you an idea of what ends up in the urine. So starting with plasma, then going on to the glomerular filtrate, and then going on to the urine, what all do we see? So sodium in plasma concentration is pretty high, it's supposed to be. It all crosses straight into the glomerular filtrate. So all of it goes into the filtrate, and some of it ends up in the urine, and the difference between the two is, of course, what goes back in also. So there's quite a bit of sodium in your urine too. Your potassium, same thing. All your potassium and your calcium and your magnesium all end up, so these are all electrolytes, including your chloride and your bicarb and all of that. These all end up for the same at the same concentration in the glomerular filtrate, but there'll be a little bit more chloride in the urine. You'll dump some calcium, you'll dump quite a bit of potassium, and you'll dump some magnesium. And then we've got some bicarb. Usually you don't lose as much as what you have, but some of it may filter out. And then sulfate and phosphate also are gotten rid of in the urine. So those are average concentrations in the urine. Substances like glucose, urea, and uric acid. So normally there's not any glucose in urine. So if you have normal plasma glucose, it will filter into the filtrate, but then it'll all be reabsorbed and you shouldn't see any in the urine. Urea, so it will leave the plasma and go into the glomerular filtrate, but a lot of it ends up making the voyage, so that you have quite a big concentration of urea, which is a waste product from protein. You have quite a bit of those into the urine. And then uric acid, same thing, it's another waste product of DNA and RNA metabolism, and it will end up with quite a bit of it in the urine. So let's see how filtration pressure is generated. So the main force that's responsible for filtration and that production of that glomerular filtrate is hydrostatic pressure of the blood here inside the glomerular capillary wall. So how is this pressure generated? Well, it's pretty cool. We've already looked at the afferent and the efferent arteriole, and you notice how I kept pointing out one's wider and the other one's narrower. Well, the fact that the efferent is narrower than the afferent causes a pressure difference. Okay, so think about the afferent bringing a lot of blood in, but then having a much smaller exit for the efferent, right? So that means that all that fluid that comes in, all the blood that comes in, can't all leave through the efferent, so it has to go somewhere. And what happens is the liquid portion is the filtrate is made by the pressure that's generated between the diameter difference between between afferent and efferent arterioles, and it pushes that fluid into the glomerular capsule. And so again, the smaller diameter of that efferent arteriole compared to the afferent raises that pressure in the glomerulus and then favors filtration. So there has to be a diameter difference between the two, right? And then, of course, you have your plasma proteins, which are too big, remember, to make it through the fenestration, and the podocytes also prevent them from leaving. So as water leaves the blood and the protein is left behind, that will increase the osmotic pressure of the blood that is inside that ball of capillaries. And then the osmotic pressure is going actually to pull water back to itself. So there are two competing forces there. And so they're illustrated here. So we have the hydrostatic pressure from that diameter difference between the two ends of this capillary, is going to push fluid out into the glomerular capsule. But then because that fluid leaves and the protein is left behind, you have that osmotic pressure that's rising. It will pull water back to itself. So it will be pulling fluid back up into the blood. And then you also have the capsular hydrostatic pressure. So that is the fact that there's fluid that enters that's being captured in that glomerular capsule. And just the presence of the fluid in that capsule generates a certain counter pressure, right? Because you can't push fluid into something if it's already full of water. So hopefully, that fluid flows, but the fact that there's fluid there can also counter the pressure here. And so generally speaking, if all goes well, you do end up with a net filtration pressure and, of course, the production of the glomerular filtrate, which will eventually become urine. And so next, let's think about this for a second. So if your kidney needs to increase its filtration rate, which of these should it constrict? The afferent arteriole or the efferent arteriole? So think about the diameter difference. Constricting means making smaller, right? It's afferent and efferent. Constricting which one would increase the filtration pressure? Think about that for a second. All right. And then the next question. If your patient's blood pressure drops to the point where the pressures in the afferent and the efferent arterioles are the same. So now we have diameters the same, pressures the same. So what would happen? Urine production will increase, it would be very dilute, or urine production will decrease and possibly stop. Which of these two scenarios would happen? Okay, so what affects the filtration rate? So the factors that affect the glomerular filtration rate, also known as GFR, are your filtration pressure, the glomerular plasma osmotic pressure, the hydrostatic pressure, and the glomerular capsule. So the three things that we looked at in that diagram that were competing forces to, hopefully to generate enough filtration pressure that it actually you get filtrate that forms. So when the afferent arteriole constricts, filtration pressure and thus filtration rates declines. So afferent, remember, it was the wide one, and then efferent is was the small one, the exit one, right? And the fact that this is way big and this is small causes pressure difference and causes filtration. If this one constricts and becomes smaller, then they're going to be equal. There won't be a difference, or it'll be very little difference, and therefore your filtration rate will decline. So and it could potentially even stop. When the efferent arteriole constricts, then, so remember you have the large afferent one here, small efferent, and if it constricts even more, then the filtration pressure would increase because the difference is even greater. So that would increase the rate of filtration. When osmotic pressure of the glomerular plasma is high, so your protein content in the plasma is high, the filtration rate would decrease because it's constantly pulling a lot of water back to itself, back to the proteins, right? And then when the hydrostatic pressure inside the glomerular capsule is high, the filtration rate will decline because that means there's already a lot of fluid in that capsule and it's going to work against more filtering in. Something that could cause that, for example, would be if you had a blockage in the tubule and the urine couldn't flow. So think of a kidney stone or something like that, and a little bitty one, right, because it would be in the tubule. If you had blockage there or a tumor or something, then that urine can't flow, and so therefore it would back up, and that particular nephron wouldn't be able to filter because there's nowhere for the fluid to go. So again, on the average, the filtration rate is 125 milliliters per minute or 180 liters in 24 hours. Most of which is reabsorbed further down the nephron. And again, we don't pee 180 liters in 24 hours. Thank God. We only pee about a couple. So how do we regulate filtration rate? So the glomerular filtration rate is relatively constant, although you have sympathetic impulses that could decrease it. So sympathetic would be like stress, types impulses. It says your sympathetic nervous system. So it's your, act, react, stress. That's sympathetic. Constriction and dilation of the afferent and efferent arterioles, of course, can help maintain peripheral resistance, systemic blood pressure, glomerular filtration rate, and blood volume. So it's trying to do a fine dance between maintaining all of those, having enough blood pressure, and really having also enough blood volume, and also filtering out. But if it has to stop urine production to maintain blood pressure and blood volume, it will. It will do that. So another control over filtration rate is the renin-angiotensin system, and that regulates sodium excretion. So let's talk about the renin-angiotensin system. So now is when we're going to talk about those juxtaglomerular cells and that juxtaglomerular apparatus. So the juxtaglomerular cells, they can secrete renin in response to 3 stimuli. Okay. Either the afferent arterial cells will sense a drop in blood pressure. So the afferent arteriole is the blood coming in. So the blood supply coming into the kidneys is limited for some reason. Okay, so there's a drop in pressure in the supply to the kidneys, and so those juxtaglomerular cells will produce renin. Or they do it because of sympathetic stimulation. So again, act, react, stress, that system. Or the macula densa cells from the distal convoluted tubule sense either a decreased chloride, decreased potassium, or decreased sodium ion content at the end of the ascending limb of the nephron loop. So there's not enough sodium that's ended up in the urine. Okay, and so it's any of those 3 stimuli will cause a release in renin. So then renin floats away into the blood, is going to trigger a series of chemical reactions that will lead to the production of angiotensin II. And I will show you in there, I have a graphic in a second that will show you. The angiotensin II then is a vasoconstrictor of the efferent arteriole, and so it will increase the filtration pressure and the glomerular filtration rate. Okay, so it constricts your efferents. Therefore, it'll increase filtration rate. That's what angiotensin II does. The presence of angiotensin II also will increase the secretion of aldosterone from your adrenal glands that sit on top of the kidneys. And aldosterone will stimulate the reabsorption of sodium. And so basically what happens is if not enough sodium is dumped in your urine, it means that you need to keep some, and you need to put it back in the blood. Okay, you can't spare a lot, and so aldosterone will make you reabsorb sodium, and as you reabsorb sodium, water is going to follow, and therefore it will increase the systemic blood pressure because it'll put more sodium and therefore more water back into the blood circulation. And the heart can also increase the filtration rate if the blood volume is high. And so this is the system that's opposite to the renin-angiotensin system, and this is the natriuretic peptide, natriuretic peptide. So natrium is sodium, so natriuretic, it makes you dump sodium, and so the atrial natriuretic peptide and the brain top natriuretic peptide, both what they will do is make you dump sodium so you can dump fluid. So it'll increase the filtration rate. So you make more urine, right? So those two are opposite each other. So your renin-angiotensin system, would it activate when you are dehydrated or fluid overloaded? Okay, so if you go back and look and think about it for a second, what does it do? What activates it? And then maybe connect the dots. Okay, so here's the graphic I was want to show you. So when the kidneys release renin, okay, there is angiotensinogen that has already been produced by the liver. So what it is this is one of the proteins you have to have in your body. It's a precursor protein. It's inactive as it is, and your liver makes sure that you have a supply of angiotensinogen in your blood just in case it needs to be activated. So renin activates angiotensinogen and cleaves it and makes it become angiotensin 1. Then angiotensin 1, through the action of angiotensin converting enzyme, or ACE, and the lung is one area where it does that. And that's one of the things also that's connected to coronavirus too, is the ACE receptors and stuff like that. You have to go back and dig into that. It's kind of interesting. But so angiotensin 1, through the action of angiotensin converting enzyme, is going to be converted to angiotensin 2. And then angiotensin 2 is going to have stimulate vasoconstriction. So you have a constriction in your vascular system, right? You're going to see an increased aldosterone secretion. Okay, so that was going to make you retain some sodium and pull water back into the system, and an increased antidiuretic hormone secretion. And we're going to talk about antidiuretic hormone, but it helps you to retain water. So to pull water out of the urine and put it back into the cardiovascular system. And it's also going to increase thirst, because obviously you need more water, because we're having to reclaim water through the action of aldosterone and the action of antidiuretic hormone, and we're having to constrict the vessels because there's not enough volume in the cardiovascular system. So all of these here are active to increase the volume in the cardiovascular system, because it was sensed that through the sensors in the juxtaglomerular apparatus, it sensed that there was either a drop in pressure, drop in volume. There was something dropped there where it needed to be compensated for. All right, so the tubular reabsorption is step number 2. We're just now talking about step number 2 of urine formation. And so all the this glomerular filtrate that is made through that filtration pressure and all of that is formed. It arrives into collecting ducts that are the collecting ducts here between the glomerular filtrate and the collecting duct. It goes through all the tubules, okay? And the action in the tubules causes the reabsorption of selected substances. So remember, in our table, we saw that all the sodium ended up in the filtrate, all the glucose, and so there's quite a bit of stuff that ends up in there that the body needs and needs to put back into your bloodstream. And so that is what tubular reabsorption is all about, is that putting the valuable stuff that made it into the filtrate that needs to really go back into the blood, putting it back into the blood. So most of the reabsorption will occur in the proximal convoluted tubules. So it'll start occurring basically as soon as the filtrate leaves the glomerular capsule and starts going down that proximal convoluted tubule. And the cells there have microvilli with carrier proteins. So remember, microvilli are finger-like projections which increase surface area, so that allows some more reabsorption, so we can get all the good stuff we need and put it back into the blood. The carrier proteins, however, do have a limited transport capacity, so any excessive amount of a substance will automatically be excreted into urine. That is also called the renal plasma threshold. So an example of renal plasma threshold is glucose. The threshold, the renal plasma threshold, is 180 milligrams per dl of glucose, which is way above normal. Okay, so if you have 180, you're probably diabetic. But anything above 180, the kidneys do not have the capacity to reclaim it, and so it will end up in the urine, and then you'll start seeing positive urine glucose in the urinalysis. So the glucose and amino acids are reabsorbed by active transport, which means you need to expend some ATP, and it's a transport molecule that's going to pull it against concentration gradients, and just going to keep pulling that substance and putting it back into the blood. Water moves by osmosis, and proteins by pinocytosis, which is again like endocytosis except of liquids. It engulfs it into the cells that line, the convoluted tubules, and then puts it back into the plasma of the blood. Other substances that are reabsorbed by active transport with limited transport capacities are creatine, lactic acid, citric acid, uric acid, ascorbic acid, which is vitamin C, by the way, phosphate, sulfate, calcium, potassium, and sodium. So again, if you reach the transport capacity, then everything else then ends up in the urine. So here it is illustrated. So you have your glomerular capsule here, and then the blood supply from the efferent arteriole is going to become the peritubular capillary. A whole bunch of good stuff ends up in this filtrate, and the body is going like, oh no, we need to put this back in the blood, this back in the blood. So we got some sodium moving back, some chloride. We have some, looks like some bicarb, right? Some water. All of that is gonna be flowing back through into the blood supply here of the peritubular capillary. So sodium ions are reabsorbed by active transport, and your negatively charged ions, which are chloride and bicarb, will follow passively because positive and negative attract each other, and these guys are buddies. So if the positive moves, the negative's got to go with them. Okay. And then water follows by osmosis. So water follows these guys too. So water likes to follow sodium. So one way again that your body has to regulate water balance is to move sodium. So wherever it moves sodium, water follows it. So if you need more water in the blood, you move sodium from the urine back into blood, and water follows. If you need less water in the blood, then you move sodium from the blood to the urine, and the water will follow, end up in the urine. So those are, it's kind of interesting. I think it's pretty cool. And then the third step is tubular secretion. So tubular secretion transports certain substances from the plasma into the renal tubule. So those are substances that may not have made it into the glomerular filtrate at the filtration station or filtration stage of the process. And so, or there's really an excess and it needs a second chance to dump it. That happens through tubular secretion. So there are some active transport mechanisms that can move any excess hydrogen and potassium ions into the renal tubule, along with other various organic compounds such as certain drug metabolites and stuff like that. So again, we have, so we're at the distal convoluted tubule here, almost going into the collecting duct, and you can have the blood here maybe has some excess potassium or some excess hydrogen ions and stuff, and what it will do in this active transport is it will actually pull sodium and dump potassium. That's what aldosterone does. Pull sodium, dump potassium. Pull sodium, dump potassium. That's what aldosterone does. And so as it pulls sodium, water follows, and then it dumps potassium. And so, yeah, that's tubular secretion. And then not illustrated here too, like I say, it could dump drug metabolites and other things. So, last chance to ditch stuff from the plasma, as at this point in time, you just about have urine. And so once it enters the collecting duct, very little happens to it, and you pretty much have urine at that point in time. So, if your patient has a blood glucose of 459 milligrams per DL, would you expect his urine glucose to be negative, slightly positive, or very positive? So again, this question relates to renal threshold of glucose. And so here are all the stages all illustrated. We have filtration that happens here, and then we have a lot of reabsorption in the proximal convoluted tubule with some secretion can happen here too. Then we go up and down the loops. There we go. And then we have very little reabsorption and more secretion here can happen also. And then we have here there's a last ditch thing that can happen here, and that would be reabsorption of water in the collecting duct can happen. And that's the last chance there. And then once it has dripped from the collecting duct into the papillae, you can't do anything to it. It's urine, no more reabsorption and stuff. Okay, so the regulation of urine concentration and volume. So aldosterone that's produced by the adrenal glands, again, from the stimulus of the renin being secreted. When it's released, it stimulates the distal convoluted tubule to reabsorb sodium ions and to secrete potassium. And then it will also, of course, water will follow the sodium. And so if you're producing a lot of aldosterone, you'll chronically have low potassium levels. You'll cramp more maybe, and then you'll also maybe retain fluid a lot. Those are all possibilities. Have high blood pressure if you have a lot of aldosterone floating around. Normally, the distal convoluted tubule and collecting ducts are impermeable to water. So normally water will not cross over. So let's say if you are plenty hydrated, you've been drinking plenty of fluids and water, and you're good to go, then the water is not reabsorbed in the distal convoluted tubule or the collecting duct. But let's say you didn't drink enough and you are dehydrated, then the antidiuretic hormone is secreted from your pituitary. If you remember, on the endocrine system, the pituitary, when it senses dehydration, it senses an increased concentration of the blood, it will secrete antidiuretic hormone, and it will also trigger thirst. And antidiuretic hormone goes straight to the kidneys, and it pokes what we call aquaporins into that distal convoluted tubule and that collecting duct, and allows water to be reabsorbed through those aquaporins and put back into the bloodstream to counter that dehydration that's going on. So again, antidiuretic hormone moves water only, whereas aldosterone moves sodium, which moves water. Antidiuretic hormone moves water only. Okay, big difference between the two. And they don't come from the same location either. Okay, so this video, I'm not gonna play it. I'm gonna send you to go watch it. I'll link it. It's a fun ASAP Science video talking about antidiuretic hormone and its effect with how it ties into alcohol and caffeine consumption and stuff. So normally I would show it in class, but if I play it right now, you won't be able to hear the sound and stuff. So I'll link it for you guys to watch. It's only a few minutes long and it's a fun little video. Okay, so let's talk about urea and uric acid excretion. So urea is a byproduct of amino acid metabolism. So if you remember, amino acids are from proteins. So basically urea is a, byproduct of protein metabolism, if you will. And then uric acid is a byproduct or waste product of nucleic acid metabolism. Nucleic acid are DNA and RNA, right? So urea is going to be passively reabsorbed by diffusion, but about 20% of urea will be excreted into urine. And then most of the uric acid is reabsorbed by active transport, and a small amount is secreted into the renal tubule and then, of course, into the urine. So urine composition will vary from time to time, so it can vary throughout the day, and it reflects the amount of water and solutes that the kidneys can eliminate to maintain homeostasis. So normally urine is 95% water and will contain urea, uric acid, a trace of amino acids, and some electrolytes. An average volume produced is anywhere from 0.6 to 2.5 liters a day. So you want to go, you want to go kind of above that to be properly hydrated and drink throughout the day. So glucose, proteins, ketones, and blood cells are normally not found in urine. So if they are present, they usually indicate a disease process of some sort, or maybe fasting if there were ketones and stuff. But those are all red flags that need to be investigated as to their cause. So if you recap this whole urine formation process, we start at the glomerulus. And we have filtration into the glomerular capsule. This is the renal corpuscle there. And then the filtrate enters the proximal convoluted tubule. Then it goes down the descending nephron loop, ascending nephron loop, and then into the distal convoluted tubule, where you have tubular reabsorption and tubular secretion that happens. These are all the renal tubule section, right? And then it goes into the collecting duct. Okay, These are all parts of a nephron right here, except for the collecting duct. And from the glomerulus to when it leaves the collecting duct are all the steps of urine formation. Again, urine formation, you cannot modify urine once it has left the collecting duct. Then from the collecting duct, it will drip through the papillae to the minor calyx, the major calyx, and then the renal pelvis, which are basically a series of funnels going from smaller to larger to the largest. And then it will drain down from the renal pelvis down to ureter into the bladder. So this is the drainage system of the kidney right there. And then the bladder will hold, so storage structure will hold the urine for a little while, and then it will leave through the urethra, which is the elimination structure. And these are all about urine excretion, or just moving it out of the body. So your ureters are muscular tubes that extend from the kidneys to the base of the urinary bladder. So they don't actually poke into the top of the bladder, they poke into the bottom of the bladder, which I thought was interesting. The wall of the ureter is composed of 3 layers, a mucus coat, a muscular coat, and an outer fibrous coat. So the mucus one being the innermost, and then muscular, and then the outer one, obviously being connective tissue to anchor it in place and stuff. The muscular coat, or the muscles, can cause muscular peristaltic waves that can push that urine to the urinary bladder, where then it will pass through a flap-like valve. Because it's going in from the bottom, there's a valve there, and you push up, and then the urine lays back down. And that's in the mucous membrane of the urinary bladder. And so that peristaltic wave is similar as the peristaltic wave that moves food through your digestive system, right? So it's basically, if you will, kind of squeezing the water, the urine through the ureter so that it can move it down. Those are also what causes pain when you have a kidney stone that's moving, because those peristaltic waves are trying to move that stone. They're squeezing around it, and it's causing pain. If anybody's ever experienced that. My daughter just had one not too long ago, my oldest one. So then the urine arrives in the urinary bladder. So the bladder is a hollow, distendable muscular organ that is in the pelvic cavity. Distendable meaning it can get bigger, it can be distended like a balloon, right? The internal floor of the bladder has the trigon, which is composed of the openings of the 2 ureters and the urethra. So it's like a trigon, a triangular area, where at each of the corners you have one of those openings. The wall of the urinary bladder is made up of 4 coats. So this one we have the inner mucus coat that has transitional epithelium, which allows it again to expand and recoil, expand and recoil. Then you have the submucosa coat, which is an additional coat from the ureters. Then you have a muscular coat that is made of what we call the detrusor muscle, and then you have the outer serous coat, which is the connective tissue, which again holds it in place, protects it, and all of that. And there's a portion of the detrusor muscle that surrounds the neck of the bladder, so where the exit is, where the trigon is, and that forms what we call the internal urethral sphincter muscles. Right? Remember, sphincter muscles open and close, right? So when they open, they let stuff out, and then they close completely, they hold stuff in. So here is an illustration. This is of a male, because you have the prostate gland illustrated here. But here's the trigon. Here's the ureters coming in. So the ureters will come in right here. Right here, there'll be a bit of a valve, flat valve. And then here's the tip of your triangle for your trigon. And you have the neck of the bladder, basically where the urethra leave. And then you have the detrusor muscle is going to be right there. And so micturition is the process of peeing, basically, the medical term for peeing. Urine leaves the bladder by the micturition reflex. And the way it happens is the detrusor muscle will contract, and the external urethral sphincter in the urogenital diaphragm has to also relax in order for urine to be able to leave the bladder. So stretching of the urinary bladder, because it's full, will trigger this micturition reflex center, and it's located in the sacral portion of the spinal cord. So it doesn't even go to your brain, although some signals will go to your brain and you will interpret it as your bladder is full. but it goes to the sacral portion of the spinal cord. So it's a reflex. So part of it is straight to the spinal cord and back. Okay. Then you have return parasympathetic impulses will cause the detrusor muscle to contract in waves. Okay. And so again, the impulses that are going to cause it to contract are parasympathetic. So you have to kind of be able to rest and relax to go pee. Okay. And then you have, when those guys fire, you do get this interpretation of that urge to urinate, to go pee. You're like, my bladder's full, I got to go to the bathroom. And when these contractions become strong enough, the internal urethral sphincter can be forced open. Okay. And the external urethral sphincter is skeletal muscle, so that is under conscious control. So basically, if you will, you have two muscles that can control the opening, right? The internal and the external sphincter. And so if it's really full and you get those impulses, that internal sphincter will be forced open, and then you can hold it with your external urethral sphincter until you're sitting on the potty. And that's conscious control. And when you're sitting, then your brain tells your bladder, okay, you can go pee, and it relaxes. Okay. And so learning to control this external urethral sphincter is what potty training is all about, getting the kids to recognize that sensation and know what to do afterwards. All right. And then, of course, the urethra is the tube that conveys the urine from the bladder to the outside via the external urethral orifice, or your pee hole, basically. It is a muscular tube with urethral glands that secrete mucus into the urethral canal. So again, there's mucus all over because, the wetness, I guess, of it all, and plus mucus protects. In females, the urethra is only 4 centimeters long, and its opening is between the vaginal opening and the clitoris. In males, the urethra serves both the urinary and the reproductive systems and will run through the prostate gland and the penis. So a short way of saying basically guys only have one hole out, is the urethra hole where both urine and semen leaves. But in females, you have the urethral opening and then you have the vaginal opening. So you have 2 holes, one for the urinary system, one for the reproductive system. And that closes the chapter. So if you have any questions, you can put them there, and we're going to talk a little bit more about water balance and acid-base balance in our next lesson.